This protocol describes key steps in the generation and optimization of efficient sgRNA vectors. A pre-selection of candidate sgRNA targets will save time and effort. The main advantage of this protocol is that it makes it possible to analyze the DNA cutting efficiencies for each of the sgRNAs without editing endogenous genes. This pre-selection method can also be applied to other gene editing methods through double-stranded breaks.
Begin by diluting the lyophilized oligonucleotides to a final concentration of 10 micromolar in double distilled water. Mix forward and reverse oligonucleotides in a thin wall PCR tube at a 1:1 ratio without adding any extra buffer. Incubate the mixture at 95 degrees Celsius for 5 minutes, then ramp down the temperature to 72 degrees Celsius for 10 minutes. Remove the oligonucleotide mix from the PCR machine and allow it to cool at room temperature. To digest the PX330-XCAS9 vector with BBS1, combine the vector, enzyme, and digestion buffer in a 50 microliter volume, and incubate the reaction at 37 degrees Celsius for 2 hours. After performing cell transformation, choose 5 to 10 bacteria.